Okay. All right. Hi, everybody. Just let everyone in here. Okay. How's everyone doing? Still breathing? Still kicking? Nah? Oh no. Yeah, I feel it too. Yeah, but today we're going to learn some more exciting biology. And I'm gonna, it's gonna be amazing. So your meh attitude will be like, woo, by the end of this, I promise. Okay, so last time we talked about a lot of things. Now you're hyped. Awesome, I love it. So, okay, last time we talked about, we began cellular respiration. We began this, let me let Sophia in. We began this catabolic biochemical series of reactions whereby we are taking our initial glucose molecule at the very beginning, right? Glycolysis, and we are breaking it down by doing these oxidation reactions and converting it into these different products in this sort of assembly line type, reaction after reaction type way, at this biochemical pathway. And so last time we finished off actually by completely oxidizing glucose into carbon dioxide. And we were generating these NADH molecules, FADH2 molecules, ATP, and of course the CO2 throughout this entire process. Okay, so continuing aerobic cellular respiration, when we did our broad overview, today is devoted to the electron transport chain. It's our last stage, electron transport chain, and then something called chemiosmosis, okay? Now, before we do that, we're gonna do a sort of fun review / warm up. Oh, I totally forgot about my announcements. Let's do that. And then we'll do our fun review warm up. So lecture quiz this week. It's gonna be on cell respiration. So everything we've been doing last lecture and then today, which we're finishing up. So that's gonna be due Friday by 11:59. And I'll email these out as well after the lecture with the recording. Write that down. Okay. And then another reminder here, journal assignment. I didn't even know some of you are working together. I think it's awesome. I'm glad I did that. My audio's acting up. Oh no. Really? Let me try something. Did I cover the mic? Can you hear me now? Oh, goody. That's never happened. Maybe my hands were on something. Is it good now, everyone? Can you hear me? Epic. Oh, good. Okay, cool. So anyway, I was just saying, I saw the discussion and some of you working together is really cool for the journal. So that, again, that's due on Friday. All right, let's get into it then. So I want to do this warm up with you guys as a sort of review of what we've been talking about. First, review the cellular respiration equation, the glucose we eat ultimately is converted. Actually, you know what? I'm going to give you guys a few minutes, and I want you to, with a piece of paper, whatever you're taking notes on, I want you to answer these questions, okay? And then in a few minutes, we're going to go over them together. But I want you to try them first, and then we'll talk about them in a few minutes, okay? Okay. Okay, let's take one more minute. 1 second.
So just kind of wait and see what happens. Oh, sorry.
Getting plumbing work done. So I asked them to keep it down. Horrible timing, but they're being quiet, so it's cool. All right, let's talk about it. So for number one, review the cellular respiration equation. The glucose we eat ultimately is converted into what molecule at the end of the Krebs cycle? So remember, we completely oxidize glucose. And you know, I'll rewrite that equation as well. So go to the whiteboard here. So our cellular respiration equation, right? we had glucose, which was C6H12O6, plus 6 oxygens, oxygen molecules going to 6 carbon dioxides and 6 water. Okay. So after learning the Krebs cycle, and I mentioned, right, that we completely oxidized glucose by the end of our Krebs cycle. What molecule was the glucose oxidized to? Not ATP. Remember what oxidation was. Okay, carbon dioxide, exactly. Exactly. So our glucose became our carbon dioxide. Remember, oxidation was the removal of these hydrogens and their electrons, right? And so glucose is ultimately going to be converted to CO2 without these hydrogens here, right? They have been stripped off glucose and they become CO2 that we exhale by the end of the Krebs cycle. Okay. So number 2, how many total molecules of NADH are produced in glycolysis and Krebs cycle combined? What was that? Not 6. So remember, we got 6 from Krebs cycle alone. 2 turns of the Krebs cycle, we got 6 NADHs total. It's close. Yeah, it is 10. It is 10. Yeah, and I'll explain why. So 10 NADH molecules. Glycolysis, we'll start with that. We got 2, right? 2 NADHs. And then Krebs cycle plus pyruvate oxidation. Did I mention that? I think I mentioned it in the question. Sorry, let me see. Yes. So pyruvate oxidation, Krebs cycle, and glycolysis. So you get 2 NADHs from glycolysis. You get 2 NADHs as well from the pyruvate oxidation step, and then 6 from Krebs cycle for a total of 10. Okay. Write a reduction reaction. So that's harder to ask on the chat, so I'll just tell you. So this was when we talked about, so it's a reduction reaction involving NAD, our electron carrier, and NADH. So if we want to reduce NAD plus, we are going to give it electrons, reducing it, and a hydrogen atom. So this would form NADH. So this would be our reduction reaction. We are reducing NAD plus. It is gaining negatively charged electrons. So we're reducing its overall charge, really. And so remember, reduction was when we would give an electron. If a molecule is being reduced, it's gaining electrons. And in biology, of course, it always comes with this hydrogen. So if you see hydrogens gained with its electrons, it has been reduced. And then, of course, the reverse of this, so the reverse reaction here, so the reverse here, that was oxidation, right? Oxidizing this molecule, you are stripping off, removing electrons and hydrogen and creating the NAD+. And so we actually saw that up here, right? The glucose lost hydrogens by the end of glycolysis, pyruvate oxidation, as well as Krebs cycle. Was this sequential oxidation reactions occurring one after the other throughout this pathway of cellular respiration. Cellular respiration, exergonic or endergonic? And why? Exergonic, yes, exactly. Why is that? What did exergonic, what did that mean? Yes, ATP is produced, released from the reaction. So it releases ATP. So we've generated some ATP so far throughout our steps here. Glycolysis got some ATP, Krebs cycle as well. But the big meat and potatoes of generating a ton of ATP we're going to talk about today, which is our electron transport chain stage of respiration. We get a lot of ATP energy from that stage. Okay, that last question here. So this is a little tricky. How many molecules of glucose would be needed to make 36 ATPs if it underwent fermentation? How many glucose molecules? If we want 36 ATP molecules. This goes back to our discussion on glucose being converted to pyruvate through glycolysis and then having that junction. 18, yes, awesome. 18 glucose molecules. And this is because remember when we did that anaerobic pathway where pyruvate was fermented to produce lactic acid or alcohol, depending on organism, right? In an anaerobic condition or environment where there's lacking of oxygen, there's no oxygen around, the glucose will be, glycolysis will occur no matter what, right? And pyruvate will be fermented. Now the pyruvate being fermented allows glycolysis to occur again with another different glucose molecule. Kind of reloads the glycolysis gun, as I sort of said. But really, the only ATP you get from that anaerobic pathway of fermentation is from glycolysis, and it's only 2. So remember that fermentation, I said it was the quick, dirty, cheap, easy way, right? Very inefficient form of energy metabolism. You're only getting 2 ATP molecules from glucose. So you gotta do it with 18 if you want 36 total. So it's 36 over 2 would give you the 18 glucose. Glucose molecules. Okay. Any questions on that? Okay. Then we can move on. So we're actually gonna do a summary of sorts of the last lecture, because of course it's a very detailed, intricate process here, as you can see. So we're gonna kind of do a review of glycolysis, or a quick summary of the overall inputs and outputs of these pathways and these reactions of glycolysis, pyruvate oxidation, sometimes remember it was called the transition step, and Krebs cycle. So let's do that. We're gonna do a glycolysis summary and a pyruvate oxidation summary. So I'm gonna set it up here. Wait, glycolysis, yeah. Okay. Pyruvate oxidation. And I'm also going to have Krebs cycle as well. Fit it into my small whiteboard here. Okay. Yeah, so 2A and 2B would be pyruvate oxidation. And Krebs cycle. And we're going to talk about the location, so where these steps occur in the cell. Get my notes out here. Let's do. So where in the cell is this occurring? Then we're going to do what's used, what is the input, and then what's produced, what is the output for each one. So input and output. Of course, sometimes there's multiple, so I'll put parentheses around an S. So I'll do that for this as well. This is one of those moments where I wish I had a bigger whiteboard, like on campus. Here we go. And then last, input. And output. Okay, where in the cell did glycolysis happen? Does glycolysis happen? It's always happening. It's only one that takes place in this area of the cell. Not the inner membrane. Haven't gotten there yet. That's gonna be later today, very soon. Not the nucleus. So glycolysis, cytoplasm. Okay, nice. Perfect. So I'm glad I'm doing this. It's a lot, and it's happening all over the place. So glycolysis occurs in the cytoplasm. So it's occurring in the fluid substance that surrounds our organelles like the mitochondria. Okay, we'll stick with glycolysis for now. What was our input for glycolysis? starting material. Glucose, absolutely. It's the very first step. What was produced from glycolysis? What was our outcome? What products? Two pyruvate, I think I saw it. 2 pyruvate, perfect. 2 pyruvate molecules, 2 NADH molecules, and 2 ATP molecules. Awesome. Okay. So pyruvate oxidation, this is when pyruvate is shuttled into the mitochondria, specifically the matrix of the mitochondria. Remember, that's the center inner area, the very center of the mitochondria. That's where pyruvate oxidation takes place. It's also where Krebs cycle takes place. It's in that matrix. Okay. So I'm going to go ahead and fill out the input here. So this is 2 pyruvate. Remember, we're just continuing along this pathway. So the product here, the 2 pyruvates, will be the input for pyruvate oxidation. For the sake of time, I'm going to just kind of go through this with you guys. But the outputs of pyruvate oxidation would be, well, does anyone remember? We'll have enough time. Two NADH, totally. You get 2 more NADHs. 2 acetyl-CoA and 2 CO2. Awesome. Awesome. 2 acetyl-CoA molecules, 2 CO2s, 2 NADHs. Great. Krebs cycle. Inputs are 2 acetyl-CoAs. A lot of outputs here, right? So we're going to get 6 NADHs, 4 CO2s, 2 FADH2s, so they're coming into play here, 2 ATPs. Woo! Isn't this fun? Yeah, lots of stuff here getting made. FADH2. This was one of our electron carrier molecules from the beginning of last lecture. Hasn't played a role until Krebs cycle. Okay. And now we continue our journey to the electron transport chain. This is our 3rd stage. So we've done 1, 2A, and 2B. We are now on our 3rd main stage of cellular respiration, aerobic cellular respiration, namely aerobic in the presence of oxygen. We haven't talked about oxygen yet, interestingly enough. This has all been without oxygen used, right? A major player. But the 3rd stage during the electron transport chain, that's when oxygen is crucial, and it's why we breathe. Okay, let's talk about it. Okay. We're still in our mitochondria, right? We kind of left off in the matrix. in the center, citric acid cycle, remember Krebs, same thing, right? And our electron transport chain occurs across the inner membrane here. And we're going to take a look at what's going on. But first, so we're still in our mitochondrion. Let's write that down. So remember, I sometimes abbreviate this as the ETC. And the location of the ETC is the inner membrane of the mitochondria. And we're going to write a couple key facts, kind of like we did with the other stages. This step yields the most ATP. So this stage yields the most ATP of all the stages. Depending on your textbook, it's roughly around 34 ATP molecules from this step. Which is pretty crazy because so far we've only gotten 2 from glycolysis and 2 from the Krebs cycle. So to get 34, the electron transport chain is very important for living. We need that energy. And like I said, the last, question. Oh, it's like an approximate. Yeah. The squiggle is like an approximation. Yeah, exactly. And the last key fact here, as I mentioned, was it requires oxygen. This stage requires oxygen. This red is starting to go on me, so I'm gonna get a different one. My roommate is telling me sorry about the plumbing thing. It's I said it's all good. Yeah, my faucet's leaking, so just adapting during COVID, right? All right. So we are going to draw this out. Yeah, fun, I know. So we're going to draw out this process, okay? So let's do it. Okay, the first thing I'm going to draw, as I mentioned, this takes place really across the inner membrane. We drew a very simplistic diagram drawing of the mitochondrion and kind of where the matrix was, the intermembrane space, the fact that it has 2 membranes, it's a double membraned organelle. And we're going to zoom in on that inner membrane, the one with the folds, the cristae. We're going to zoom in. So we have this lipid bilayer, right? A phospholipid bilayer, which is going to extend out, just like all our cell membranes, right? And Above, in our drawing here, would be the intermembrane space. And below, center of the mitochondrion, so that's the matrix. Okay. So I'm going to draw some, the screen is blurry. Oh, I see that. Let me move it up a little bit. Okay, I'm going to draw in some key proteins. Now, remember when we learned about the membrane, there are proteins embedded in membranes. These are so-called membrane proteins. So certain transporters, channels, things like that. So I'm going to draw some in. Let me draw one here, here, here. And then this last one looks kind of funky. Make it a different color because it has a different function. Okay. So this whole process has kind of been a little elusive to you in the sense of why are we creating or oxidizing glucose to generate these full taxi cabs. Remember, it was kind of that taxi cab analogy of the electrons were our passengers in these coenzyme NADH and FADH2 molecules. And really, you're probably wondering what the point of doing that is. What's the point of filling these empty taxis with electrons from ultimately glucose and creating these NADHs from NAD+, FADH2s from FAD? It's here, the electron transport chain. This is sort of the destination for those taxis. They're picking up their passengers, these electrons and hydrogens, and dropping them off at the electron transport chain. So we're going to start here with NADH. And of course, we have 10, right? But we're going to just do one just to show the process. So let's do NADH. And yes, Glorianne, so the purple squares are proteins. Yeah, in fact, let me label them. So these are membrane proteins. We'll label this guy soon. So NADH is going to drop off its electrons to the electron transport chain. If I strip off electrons from NADH, am I oxidizing or reducing NADH? If I'm converting NADH to NAD+. I'm oxidizing it, absolutely. I'm oxidizing NADH. And so the electrons from the NADH So here's our oxidation reaction. This is oxidation. These electrons are going to be picked up by this membrane protein. They're going to attach to that membrane protein. Like that. And you know what? I'm actually going to draw them a little higher. I'll tell you why. Because these electrons that are dropped off from NADH are in a high energy state at the very beginning of this chain. So what's happening here is that high energy electrons here at the beginning of this chain are going to move down from protein to protein, down our electron transport chain. So let's just do one. One transfer. So these electrons are going to go to this. Now notice I drew it a little lower. Each transfer here involves a loss of energy, meaning the electrons here are at a high energy state, and then the transfer here, they go to a lower energy state. Now, if you remember from the very beginning of this unit, we talked about the first law of thermodynamics. Energy cannot be created or destroyed. It can only be transferred or converted from one form to another. So, if my electrons moved, if they had a lot of energy here and they lost some of that energy, it doesn't just disappear. We can actually harness it. The cell can actually use that energy. And what it does, these membrane proteins are actually proton pumps. They are hydrogen ion pumps. And so with the energy that is lost, it's going to pump with that energy that's conserved, right, and available now. It's going to pump hydrogen ions across the membrane. And this protein will do it too. The electrons will lose more energy here. That energy will be used to pump these H pluses across from the matrix to the intermembrane space, and it starts to build up. In fact, it already was built up. We are moving from an area of low concentration to high. This was my suitcase analogy. Do you guys remember this? Do you remember what kind of transport this is? When you go from an area of low concentration to high? What? I know this was a few lectures back. What form of transport required energy? Active transport. Totally. That's exactly right. And active transport was the only form of transport where you're moving from a low concentration, so there's a low H plus concentration, right? Those are the brackets, to high H plus. This is the analogy I used with the suitcase where it's starting to get really full. There's a lot of stuff in your suitcase and you want to add more. You're going from low to high. It requires energy input. These membrane proteins are acting as pumps to pump up hydrogen ions against their concentration gradient, going from low to high. That's an energy requiring process. And that energy comes from moving electrons from a higher energy state to lower. If you go from higher, if that electron lost energy, went from higher to lower, that energy doesn't disappear in the universe. You can use it to do an energy requiring process, such as active transport. Pretty amazing. And that's what's going on here. I'll quickly add, while we're on the topic of electrons being dropped off and moving down the chain, FADH2, very similar process, except it's oxidized, but its electrons actually are picked up by the second membrane protein. They just start there. So let me do FAD plus there are the electrons. They just go immediately. Probably should have did a different color, but they go immediately to that second protein and do the same thing. And this is plus 2 hydrogens, right? FADH2 to FAD. It's definitely one of those lectures I wish I had a large whiteboard. Oh, I just heard the plumber. He's done. Okay. Next part of our story is our final funky looking protein here. This protein is called, quite appropriately, ATP synthase. So this enzyme is called ATP synthase. It's a pretty amazing protein. In fact, it's so important and amazing that its function and structure and the way it catalyzes this next reaction won scientists who discovered it the Nobel Prize. Peter Mitchell, I believe his name was, I think. But yeah, so you'll see why it's amazing, and it's happening in all of our cells, and it's like, really? Wow, pretty cool. Okay, so continuing our story here, as our hydrogen ions are being built up in the inner membrane space, we learned when we talked about transport that molecules typically, without energy input, they tend to diffuse from high to low, high concentration to low. That just happens by way of the universe. It's a more stable situation. They want to move from high to low. It doesn't require energy input. And that's what's going to happen here. As we have our high hydrogen ion being built up, right, by our membrane proteins and the electrons moving down, pumping the H plus from the matrix up, they're going to want to move down. And they do. They do over here. They start to flow through ATP synthase. They actually bind to it and then unattach and be released down here. But the amazing thing about this protein is that as hydrogen ions are moving through ATP synthase, it actually causes it to spin like a rotor on a boat. As it moves and water moves through past the rotor and it spins, right? This protein spins around, spinning around. And what's happening, amazingly, is as it spins, it is phosphorylating ADP. We drew out ATP and we showed that high energy bond, right? So it is taking ADP with 2 phosphates and it is attaching on a phosphate, a third phosphate, continuously. Just slapping on phosphates as it spins around, just like this, as hydrogens move through, okay? It's churning them out like a machine in our cells, doing or taking ADP plus phosphate, inorganic phosphate, And I'll move the whiteboard closer in a minute. And creating, let's do an arrow here, ATP. And lots of them. About 34 per molecule of glucose. Right here. Okay. It's a long explanation, long story, but there's one more thing. What's the squiggle next to P? Oh, it's not a P, it's a plus, sorry. So ADP plus phosphate. To generate ATP. Okay. There's one more part of the story. What am I forgetting? What haven't I talked about yet that I told you I'd talk about? Oxygen. Where's oxygen? What role does oxygen play here? Let's get another color. Phosphorylating. No. So, okay, oxygen. That's the one I haven't talked about. And you know, something else I didn't talk about is, and we're wrapping up our story, really. This is the end of this entire process. I haven't mentioned the end products. We've talked about CO2. We covered that, right? All 6 of them. But water as well is produced at the end of all this. And I'm going to show you how. Oxygen, doing green, actually binds to this final protein in the chain. So O2. And it acts as this final electron acceptor. So final electron acceptor. Remember, we learned in chemistry, the chemistry unit, about the electronegativity of oxygen. The fact that oxygen loves electrons, has this very powerful attractive force for electrons. And he would hog them in water, right? That's actually why these electrons move closer and closer down to oxygen at the end of the chain. Oxygen is exerting its influence on these electrons and pulling it towards it. It loves electrons. It has that electric attraction for them. And so that as they move down, and they'll finally reach him. They'll finally reach him and bind to him. And that's where water comes in. So once oxygen combines with the electrons, it's going to become even more stable in this universe. That's things want to become more stable. It's going to attach on 2 of the hydrogens down here. So oxygen actually becomes reduced to water. And so it actually looks like this. 1/2, so oxygen splits. Let me erase here. So 1/2 oxygen plus 2 hydrogens. Just write this beneath on your paper. Because I gotta make some more room here. To form a water molecule. 2 H's, right? H2O. So yes, oxygen is reduced, right? We added hydrogens. We also added electrons, right? So we reduce oxygen to water. And this is actually why on a cold day, you don't just breathe out CO2, you're breathing out water constantly, even now. But on a cold day, if you go up to the window, you can actually see the water vapor condense on the window from this, leaving your cells. So at the end of all this, what do we get? ATP and water. And we've oxidized NADH and FADH2 back to NAD and FAD, which is what we started with in glycolysis. Therefore, we can go back and do the whole thing again. Make even more energy. So wasn't that totally not complex and insane? It took me a long time to learn this, so it's a lot. There's no doubt about it. Any questions? Okay. So while this is up, question. So the NADH and FADH2 are recycled. Yeah, that's exactly right. They are. Yeah. They're oxidized, and so they can be used again. Totally. Are the O2 always on the last? Yeah. So oxygen is actually going to bind to that last protein because it's pulling these electrons down the chain. At least that's what the textbook says, and it's sort of the prevailing theory. I think there's actually still active research on this process. It's very complex, as you can see, but fascinating. And I during grad school, I actually was very interested in this researcher who was looking at cancer cells that were thought to be defective in their ability to do this process. It was really interesting. End products are ATP and water. Yeah. And if you want to include NAD plus and FAD, you can, of course. Those are also going to be generated at the end because you're removing their hydrogens and their electrons. In fact, maybe that's what I'll do because we have some time. I'm just going to draw out the whole thing. So let's do a similar sort of thing that we did with the other processes, with the input output. I think that'll help your notes and kind of keep it all consistent. But before I do that, I'll mention this is the electron transport chain, right? As electrons are moving down this chain of proteins. And then, it's a question. Yeah, it's half. It's literally half. The oxygen O2 splits and joins up with 2 hydrogens. And the other oxygen from it can join up with 2 other hydrogens to form another water molecule. Okay, so this was our electron transport chain. But while this is up, I want to mention, and we're going to write this again in a moment. This, I'll answer that yes. So let me answer that question in a moment. So this part of the story, this part where the hydrogens are naturally simple diffusion, facilitated diffusion, right? There's a protein involved going from a low or a high concentration of hydrogen ion to low concentration of hydrogen ions. We call this process chemiosmosis. Just has a fancy name, but it's just referring to the fact that these hydrogen ions are moving through ATP synthase to an area of lower concentration in the matrix. That's chemiosmosis. This is our electron transport chain, and we have, so part of this third stage, it's the electron transport chain with and chemiosmosis. I'm actually going to write that so you have the spelling. And in my slide too, it's on the next. And there's another fancy term as well that we'll talk about. But okay, let me answer that question. Is it supposed to be 1/2? It is 1/2, yeah, so I'll type it in. 1/2. Okay, let me see here. Okay, so we just did that. Do I not? Okay, never mind. Okay. Okay, so let's see. ATP synthase. So here's this process here, right? Of hydrogen ions going across the membrane and ATP being formed, catalyzing this reaction of ADP and a phosphate group coming together, forming ATP. So we're going to write some stuff about that. So you have that word chemiosmosis. I just don't want to erase this because it's so beautiful, but it must go. Okay. So ATP synthase. Let's write about it. It's the enzyme in the electron transport chain, or at the end of it, end of this stage, that catalyzes the formation of ATP. So it's combining ADP molecules with a phosphate to produce ATPs. It is, Anastasia, yeah, it is part of this overall third stage, the electron transport chain. And it's this enzyme at the very end, yeah. Oftentimes, electron transport chain and this last part, chemiosmosis, is kind of coupled together in this third stage of respiration. So it requires a proton gradient, those 8 hydrogen ions, those positive hydrogen ions, right? Requires a proton gradient. And let's write that fancy word. So the protons that pump through ATP synthase, or pump's not the best word, I should edit that in my notes, that move through ATP synthase move by a process called chemiosmosis. Just the name for this movement. It's a chemical, technically, right? Moved by a process called chemiosmosis. Okay. One more fancy term that you can throw around at your dinner party later, your socially distant dinner party: oxidative phosphorylation. We're going to write that. So this overall electron transport chain with chemiosmosis, creating ATP molecules. Sometimes it's referred to as an oxidative phosphorylation. And what that really means is by oxidizing NADH to NAD plus, FADH2 to FAD, it's generating the power to phosphorylate or stick a phosphate, transfer a phosphate group to ADP. Oh, cool. I got a cool message. Okay. Cool. All right. Yeah, it's an awesome word. 2 words. Okay. So let's write it. Let's define it. So like I said, the ETC and chemiosmosis is a process of oxidative phosphorylation. Let's see if this red's better. Oxidative phosphorylation. And this is when, this red is no bueno either. It's good enough, I guess. This is when ATP, oh, this red's great. Cool. Switched it out. Okay. ATP is generated from the oxidation, ultimately from the oxidation of NADH and FADH2. These full taxi electron carrier molecules being oxidized, dropping off their passengers at the electron transport chain to subsequently move down allowing for this pumping of protons across up into the intermembrane space against their gradient, against their concentration gradient, which of course requires energy. It's active transport mechanism with these proton pumps in the membrane, which then flow down ATP synthase from high to low just by facilitated diffusion, no energy input required, generating ATP molecules by this spinning mechanism in the ATP synthase molecule enzyme. And lastly, oxygen is required. It's at the end of this chain, pulling these electrons, and when they reach oxygen, oxygen will combine with two hydrogens to form water, and it's reduced to water. Okay, so with our last 4 minutes, let's do that sort of input-output summary of our third stage, the ETC. So where in the cell? Right? And then the input and outputs. Oh, I have a lot more room. Outputs. So where in the cell? Let's see if you guys help me fill this out here. Where is this happening? Mitochondrial inner membrane, totally. So the inner membrane of the mitochondria. What was our input or inputs? What did we start with here? I'll help you out. Start with NADH, right? 10 NADHs to be accurate here, right? From before, from our earlier processes of creating them. Yes, 10 NADH, 2 FADH2s, and of course oxygen, right? So I'll write O2, which is oxygen gas. So that's our input. That's what's required to do this. And what's produced ultimately, 10 NAD pluses, because they're oxidized, right? 2 FADs, again oxidized, and oxygen is reduced to water, H2O, or water. And then of course, the most important thing, can't forget, our ATP, right? So let's write roughly 34 ATP molecules. Okay, and then I'm actually gonna write 34 ATP per glucose. One glucose molecule throughout this entire aerobic cellular respiration is going to ultimately, with glycolysis and with Krebs cycle, because no ATP from pyruvate oxidation. Krebs cycle, glycolysis, and this ETC stage with chemiosmosis, we're going to get 36 to 38 ATPs, okay? But 34 from the ETC specifically. The other 4 come from the other stages. So I'm going to write ATP per glucose. It can vary depending on the organism, but I just want you guys to know roughly 34 from here, 2 from glycolysis, of course, and then 2 from Krebs cycle. 34 from electron transport chain. Okay, how are we doing? Okay, minute over, we're all good. So any final questions here before we take a 10-minute break? Maybe we need a 15-minute today after that. I'll stand here. And this, by the way, says per glucose. So we started with 1 glucose molecule. Yes, Glorianne, that's exactly right. So we got 2 ATPs from glycolysis and 2 from Krebs. So that would be 4, and then 34 from here, so 38, sometimes 36, depending on the organism. Okay, so you guys 10-minute or 15 today? It's 4:17 before we 15 immediately. I'm okay with that. So you guys want to do a 15-minute break, come back at, let's do a hybrid. Let's come back at 4:30. So that's like 13 minutes. 4:30 for the lab lecture. Good? Okay, awesome. So 4:30, we'll talk about microscopes. It's a good song.